Okay, ayan. So, hello dears. Okay, so, ayan. So, we'll now have another pre-recorded lecture in your bacteriology class. Okay, and this time, we're now going to talk about the third specimen na ito i-process, which is stool. Yes, atong bestie, sukad second year. Your attach, okay? Stool, okay. So we have already discussed from the previous lecture the different routine culture media na itong ginagamit for stool culture, okay? And before we proceed, na ko yung mga additional na mga notes. Alright, so mnemonics na pud ni, okay, di ba? So for those culture media daw na ay neutral red, okay? Neutral red na pH indicator, unsa to ang mnemonic? SMAC, okay? Do not be confused aning SMAC with sorbitol McConkey ha, lahi ni siya. Ako na lang SSMAC. Okay? Alright. So because ang naay mga neutral red, you have SSA and McConkey. So sila lang duha ka culture media ang naay neutral red ang pH indicator. Okay? Another one is kung phenol red. Okay? Phenol red. Kung phenol red na mga na pH indicator, kinsa din mga culture media, you have mix cut. Okay? MXCUT. So sa meaning sa mix cut, M stands for MSA. X stands for XLB, C stands for CTA, U stands for urease, okay? And T stands for, oh my gosh, alam ko sa meaning sa T, TSI. Okay, ayan. So, basically, kanina mga mnemonics is overall na siya na mga culture media, yun natong naagian. Okay, so kung mixed cut, MSA, XLD, CTA, or cysteine trypticase, cysteine trypticase agar ba ni? Yeah, CTA. And urease, letter U and T for TSI. Okay, that's for phenol red, mixed cut. Okay, and next, last two indicators, you have bromthymol blue. Okay, bromthymol blue. Unsa to mnemonic ani? CHOT. Okay, CHOT. So C stands for citrate. H stands for hectoen enteric. Okay, enteric. O stands for OF glucose. And T stands for, what's the T? Bromthymol blue. T, OF glucose, T, o diba nakilala ito na po sa TCBS. Ano ba yan, Mark? TCBS. Okay, CHOT, bromthymol blue, citrate, HE, OF glucose, and TCBS. Okay, and the last na to na indicator is, of course, you have your bromcresol purple, okay? Bromcresol purple, you have, di ba kato, LIA. And kato mga ornithine, arginine, di ba? So kato mga, usually for decarboxylation tests, your bromcresol purple. Okay. Alright, so ayan. So those are just some mnemonics, okay? And another thing po di ba, we have BSA. It's for, specifically jud for Salmonella typhi, okay? Ang imuhang bismuth sulfite agar. Now, we also have your brilliant green agar, okay? Brilliant green agar, it's used for the other Salmonella species except na ni S. typhi, okay? So, kung S. typhi ang pangitaon, BSA, bismuth sulfite agar, and kung the other Salmonella species na brilliant green agar. Tama ba, Mark? Okay, ako lang taon ako ang handout sa ako ang review sa una. Okay. Alright, okay. Sige. Okay, tama. So, bismuth sulfite agar, it's for Salmonella typhi, and brilliant green agar is for the rest of the Salmonella. So, sa brilliant green agar, guys, dili na ma-detect ang Salmonella species. Salmonella typhi. Bismuth sulfite yun ang makadetect sa imuhang Salmonella, okay, tama. So, ang bismuth sulfite agar, it's specifically good for Salmonella typhi na isolation. Ang brilliant green agar, okay, brilliant green agar is for the other Salmonella species except this time S. typhi. So, sa brilliant green agar, dili na ma-detect ang S. typhi. Okay, so kanina mga additional na to siya, no? Mga mnemonics na baka lumabas sa inyong exams puhon or sa board. So, at least na namoy ano. Alright. Nanamoy sa na initial na mga thoughts or whatever. Okay, alright. So we'll now start with stool culture. So how do we process stool? Okay, so again, this is our bestie, so pag second year. So we'll now talk how do we culture stool, okay? And what are the pathogens na itong pwedeng makita from stool. Okay, so we'll start first again with stool. Again, it's a specimen of choice for our GI pathogens. Excuse me. Of course, because it's stool, therefore, atong gine-detect is the gastrointestinal pathogens. So those that affect the GI system. So basically, katong makapadiarrhea, maka-gastroenteritis, etc. Alright? Okay, and it's one of the most commonly encountered specimen, ang stool na to. Especially, not only in bacte or micro, but also in CM, right? Especially for para. It's one of the mga specimens na itong ma-encounter and one of the specimens na medyo kinakatakutan or mga inaayawan. Yes, okay, the stool specimen. And of course, for rectal swab, our discussion from the previous lecture on swab specimens, again, it's not ideal and it's not sensitive also to detect gastrointestinal pathogens. And it can only be used in certain populations, usually for infants and small children. Because these people or these patients, they do not necessarily or they do not usually say na, uy, malibang na ko, or hindi sila makasulti ba na malibang na sila. So, we use rectal swabs kaya para makuha dyan nato itong stool or wala tayong ma-miss na specimen in a way. Okay? Alright. Pero ang specimen of choice dyan, if you want to detect gastrointestinal diseases or to detect gastrointestinal pathogens, is always stool. And di ba rectal swab, recall, rectal swab, mag-rectal swab mangani ka, dapat visible good ang feces. Dapat naitain na ma-appeal sa swab. Okay? Alright, ayan. Now for our stool specimen, same with your para, no? Pea-size ra, pea-size. And if liquid, about 5 ml. Or, di ba sa inyong para, I think 10 ml ba to? 10 cc of liquid stool. And dapat placed in a dry, clean, leak-proof container. Di ba, as you can see, clean ra ang gibutang, not necessarily sterile. Because again, your stool already contains a lot of normal flora, normal mga enterics. Okay, but of course, since we're looking for culture, pwede po na mag-sterile ta. But a clean, dry, leak-proof container will suffice. Okay? Alright. Ayan. Now, again, it's best to collect the fecal sample during the acute phase. When you say acute phase, sugod pa lang sa infection. So, para at least, if mo collect ka during the acute phase, you will ensure that makuha dyan mo itong pathogens at its peak or like kanang daghan pa sila because sugod pa man og infection. Okay? So, wala pa'y tambal, dili pa dugay. So, medyo fresh pag yun, daghan pang pathogens na present sa imuhang specimen. So, it's best to collect it at the acute phase of the infection. Alright. And collect fecal sample directly into the container. Of course, that's imperative, especially if it's culture. Culture? Puberty, guys. Oh my God. Sorry, okay. So we have to put it directly into the container because we don't want, of course, external contaminants that could hinder or that could join or that could affect our culture results, especially culture ang atong buhaton. It's much possible dapat na tanan is sterile, tanan limpyo, tanan free of contaminants. Okay? Alright. And again, as mentioned, sample must be free from contaminants such as urine, of course, because urine has its normal flora, or if ang patient po na ay laing sakit na urine ang specimen, so pwede po siya maka-introduce og laing kagaw si mong tae, yeah? Barium and of course toilet paper. Saan naman ko eh? Toilet paper. Alright. So of course, because again, these can introduce contaminants. Alright. And of course, label as always, di ba? So, oh my God, hapit na mo man ang first sem, hapit na po mo man ang third year. Kailan pa ba emphasize ni? Yes, mga medtech dapat, of course, gasgas na gasgas. Dapat label good ta sige. Ha, dapat na label. Naku, sinasabi ko talaga. Alright, ayan. And of course, dapat patient name and 2 identifiers. Usually, time, the name and the birthday and hospital ID would suffice. Pero usually, pwede na name and birthday, okay na siya, or age ba. And of course, time and date of collection din. Alright? Okay. Now, for rectal swab, if there are rectal swabs na tumapil, again, must be approximately 1 inch. The inch or diri, pero diba, about 2 cm to siya. Insert siya beyond the anal sphincter. Okay, and then atong i-rotate. And as much as possible, again, dapat na ay visible na feces on the swab. Okay? Alright. And, of course, as mentioned ako sa lecture on swab specimens, most of your swab specimens, if not all, if ato magaling siyang i-culture, okay, they are usually put in a transport medium. Because again, most of the situations, manggod, your patients are far from the laboratory or far from the testing site. So, it would take time pa to travel from the point kung asa siya gi-collect to the point of inoculation or to the point of culture. So kato siya na travel time that could play a significant role na in our isolation. Because kato siya na time, pwede na to siya makakause of deterioration sa imong organisms. So as much as possible, we want to prohibit that or we want to avoid that. That is why ato ibutang ato ang mga swabs in transport medium para at least sa on the way pa lang sa lab ma-preserve ilang morphology, dili kay ma-inhibit ilang growth muna, okay, before para dili sila mo overgrow kaayo, hindi po sila mamatay dayon. Okay? All right. Ayan. So, again, there are two types of your specimens na stool could be unpreserved or preserved. So, kung unpreserved, like fresh, it should be processed within 1 hour. Okay? Within 1 hour dapat. So, dapat mahatod siya sa lab and dapat i-process na siya sa medtech within 1 hour. And if preserved siya, example, sa swab siya gikan, or like nabutangan siya preservative, for as much as possible for bacteria or for culture, di gyud nato nabutangan og like liquid preservative like formalin ba or something. So, if preserved siya, it's viable within 24 to 48 hours at room temperature in holding media. Now, let's say lang, assign ka og assign kag CM, ay clean mic na section sa lab. Nakadaot kag stool, okay? And then ang request niya is na culture and then napoy kanang DFS or direct fecal smear. So asa man imong unahon ana? Of course, if ikaw makadawat kag specimen, unahon jud mo na sa micro. Okay? Unahon jud mo ang micro because again, we want to isolate as much as possible the organism as fresh or as less deteriorated as possible or as early as possible. Because again, if unahon nimo ang imuhang example, mag-una kag DFS. So pwede na siya ma-contaminate, mapadugay na siya ang imuhang stool na unpreserved pag yun kay gi-process pa nimong DFS na kanang pag-determine og parasite. Nya na day culture dito, so wa na ni overgrow na itong mga kagaw or could be namatay na itong important na pathogen dito. So gani if nakay mga specimens, not only stool but other mga fluids or mga blood whatever na usually ma-share siya within how many sections sa lab. If na gani micro, always prioritize micro. Okay? Siya jud ang priority. Because again, especially good mga body fluids kanang example na apay culture and then napo siya histo na kanang gamaan og cell block like ibutan nimo siya og kanang paraffin and all that nya gamaan nimo ang cell block then butang ka man og formalin di of course die if nauna na kag histo nya napay micro na culture gibutan nimo formalin na nga matay na ang kagaw tanan wa na kay ma-isolate. All right, so mga inana na mga situations guys that's really common to encounter in your internship and very dangerous, very risky pod na di ka magdinangag mga inana na situations because again pwede kang ma-demerit, pwede kang ma-reprimand and all that. So di dyan magtanga-tanga ha. Okay, pero naman yung mga inana na situations or anang mga experiences. I'm not saying na ako, I think pero pero as much as possible dyan we have to avoid. Okay, so mura na akong point na if you have specimens na kanang pwede ma-share ba sa other sections sa lab, like pwede siyang micro, pwede siyang clean chem, pwede siyang hema, pwede siyang histopath. So, always prioritize good micro. But that would depend on the laboratory. But most of the time na ako na-experience, prioritize good ang micro because you want to isolate man. You want to culture. So, if culture man ganyan, ang imo na yung mindset ana is you have to make sure that it's really free or ma-minimize ang contamination. Okay? Because para dili gyud ta ka maka-misdiagnose. And ma-plate na to siya as early as possible para dili ma-deteriorate or dili kayo daghan or ma-minimize ang loss of organisms. Okay? Alright. Dami kong chika. Alright. So that's a different types of stool, unpreserved or preserved. Okay. Now for transport media, we can always use a lot of transport media. But ang pinaka-common is your Kerry-Blair medium. And we could modify that. And one of its modification is atong increase iyahang concentration from 0.5 to 0.16. And it's usually used for Campylobacter species. You could also use alkaline peptone water. Kakinsa man alkaline peptone water, it's for Vibrio cholerae. Now, I'm not sure if pwede ba siya i-transport, but si Sir Richard ba nagamaani? So, yeah, I think it could also be used for transport. Pero alkaline peptone water is also used for enrichment. Okay? So, it's enrichment broth, pagpadaghan. Okay? Sa imuhang Vibrio cholerae. Okay. And 1:1 glycerol and 0.033 molar phosphate buffer. This is for Shigella species. And you could also use Stewart's and Amy's medium. All right. But most of the time, if stool specimen, ako yung ma-encounter if transport medium is Kerry-Blair. Okay? Kerry-Blair medium. All right. Now, for the stool culture procedure, Of course, what we do lang is, you receive your stool, examine it macroscopically, macroscopically. So same as sa parasites, sa atong parasitology na class. You examine the color, you examine the appearance, and you look for kani mga bloody or mucoid spots because it is in those na areas na medyo concentrated ang kagaw nato. Alright? And if sa para, napudra ato ang mga ihis or baby jarja ba, whatever. So usually dito na makitaan. Alright? So what we do is we use a cotton swab, okay? Cotton swab. Alright? Sterile cotton swab ha. And then ato na siyang i-poke or i-tease. Oh, what a tease. Lahi ng tease na yung nahibalaan ha. Alright. So sterile loop or swab. Again, i-tease siya at different areas. And especially prioritizing these areas na may bloody or mucoid spots. So dapat ma-appeal na siya sa pag-tease. Alright? And then after, ato na siyang inoculate sa different primary plating media nato. So same na siya sa atong swab specimens na pag-inoculate. Ang swab mo siyang i-roll sa first quadrant. Okay? And then after, mag-multiple interrupted na ka using a loop until the fourth quadrant. Okay? Alright. So same na siya og streaking pattern sa imuhang swab specimens. Okay? Alright. And again, these are the different primary plating media for stool. Di ba na-mention na nato for last lecture na to on the routine stool culture media. Alright? So of course, makonki or AMB because again, it generally isolates your gram-neg enterics. Okay? Alright? Wala silang pili. Basta gram-neg enterics. And of course, we have for isolation of Shigella and Salmonella, pwedeng SSA, pwedeng XLD, pwedeng HE, di ba? Alright? Media for isolation of Shiga toxin or your EHEC, E. coli 157H7, your, of course, sorbitol maconchi. Isolation of Campylobacter because again, these are enteric, non-enteric pathogens. Okay, dito sila part sa normal flora sa enteric, but they can cause diseases sa imong enteric or GI system. So Campylobacter, CCDA, charcoal cephaloperazone, deoxycholate agar or Campi blood. And additional plates, blood agar if na gram-positive organisms, especially for Clostridium na mga Clostridium difficile or difficile na infections. TCBS again for Vibrio if suspected na Vibrio case. Or CIN, di ba? Cephsuludin ergazan, Novobiocin agar for Yersinia and Aeromonas. Okay, so that's for the stool culture procedure. Again, for streaking patterns, same ra siya with swab specimens. Now, you roll the swab on the first quadrant, and then after rolling, you use the inoculating loop, okay, streak ka until the fourth quadrant using MIS or multiple interrupted. Okay? Alright. Ayan. Okay. And you incubate plates again at normal incubation temperature for about 48 hours. Pero kung you're expecting for Campylobacter, you incubate at 42 degrees Celsius and microaerophilic environment, example, candle jar. Because again, recall that your Campylobacter likes to grow at 42 degrees Celsius. Silang duha ni Helicobacter, di ba? And also Pseudomonas, pwede pod. But again, Campylobacter really grows well at 42 degrees Celsius. So you put it in an environment na 42 degrees Celsius and also microaerophilic or decreased or lessened amount of oxygen. Okay, alright. Ayan. And enrichment broth also may be used samot na if gamay pa kayo ang pathogens na naa. So you could use again APW for Vibrio or kung mga enterics pwedeng selenite F, okay, or for Salmonella tetrathionate. Okay, later. We'll discuss that. Okay. Alright. Examine plates at 24 and 48 hours and isolate single colonies with distinct morphology. So katudyo mga distinct. So especially pwede lactose fermenters, distinctly colored colonies o mga non-lactose fermenters. Usually for for enteric na mga diseases like gastroenteritis or mga diarrhea, what we're really looking after is katudyong mga true pathogens. Okay, so example, Salmonella, Shigella, Yersinia, you have Campylobacter also. You have Vibrio, E. coli 157H7. And Salmonella Shigella, for example, on maconchi, those are, of course, non-lactose fermenters. So if ikaw medtech in the future, makakita ka, MAC, yan ay mga non-lactose fermenters, could be the first imuhang priority na i-isolate yun is kato mga non-lactose fermenters. Because at the back of your mind, that could be Salmonella or Shigella. But then again, pwede silang late lactose fermenters pod, which are just normal commensals or normal flora. So, pwede pong i-disregard siya. So, it's important that you are really familiar with the different colonial morphologies and colonial characteristics of your pathogens. Okay? Alright, ayan. Okay, and again, perform appropriate workup. So, when you say workup, like, if kanya akong isolate, okay, kitaan ko non-lactose fermenters, colorless. So, unsa akong next button? Unsa na test perform? Mag-PSI ba ka? Unsa ba ka? Citrate, etc., etc. Until makabot ka sa imong identification or identity of the organism. Okay. Alright. And take note, if walay bacteria na ma-isolate, collect 2 more samples at different time points. Because again, there are some etiologic agents that may be shed intermittently. When you say intermittently, not present at all times or not always found in fecal material. So, naay specific time kung asa siya makitaan. Okay? Alright, ayan. And last note, actually ako nang siyang i-include from Bailey and Scott's na table, di ba? Naka-mention ko about chapter 5 sa inyong Bailey and Scott's. Nag-summarize na niya tanang specimens and their specifications, unsay pag-process, etc. Unsay culture media kailangan. So ako na siyang gi-appeal. Alright, so ang yang note dito is do not perform routine stool cultures for patients whose length of stay in the hospital exceeds 3 days. And ang yang admitting diagnosis was not diarrhea because later we'll know about, because again, they are suspected na of being infected with Clostridium difficile or difficile. Because remember, wala siya na-admit og diarrhea. Okay? Iyahang initial diagnosis was not diarrhea. But then, after 3 days, nagyapon siya sa hospital niya, nagkasugod na siya kalibanga. That could mean na naana siya infections or secondary infections caused by antibiotic-associated na colitis. And that is caused by Clostridium difficile. We'll know later sa atong lecture. Okay, kalma lang. Alright? Okay. Ayan, so just some reminders. Okay. And now we go now to some terminologies. Of course, for enteric bacteria, when you say bacteria, enteric bacteria, they are again bacteria that are recoverable from your intestinal tract, the normal flora of your GI system. Enteric pathogens are the pathogens that causes disease in the enteric system or your GI system. Coliforms are another term for lactose fermenters. And paracolon are your late lactose fermenters, so mga just terminologies. Non-lactose fermenter, of course, nasa pangalan na, they do not ferment lactose. Non-fermenter is they do not utilize sugars do not ferment carbohydrates, but they either oxidize, which is an oxidizer ang term, or dili gyud sila mogamit og any sugars or carbohydrates, which is ang atong term is inert or asaccharolitic. Okay, so just some terminologies, I'm sure you're all familiar naman pod. Self-explanatory naman din. So just to guide you siguro when you read mga textbooks puhon or mga articles ba, yeah. So those are the meaning of that. Okay, and for the next video, part 2 of our stool culture, we'll now look into the different pathogens that we may isolate from stool. Okay, so we'll start first with Salmonella, Shigella, Yersinia, E. coli, Vibrio, Campylobacter, and last is Clostridium difficile. Okay.